Hi, I'm Steve Gilson from the Alliance for Southern California Innovation. We are a regional super connector. Welcome to Research Innovation Spotlight series on genomic sequencing. Our great moderator today is Michael Hultner. He is currently the SVP of Corporate Development at Valsera. He supports the Chief Corporate Development Officer and is responsible for corporate strategy, M&A, and strategic partnerships in precision medicine. Prior to Valsera, Michael was VP and Head of Genomics Strategy at United Health Group, where he contributed to the genomics and precision medicine strategy of Optum and UHG. He founded Optum Genomics and served as GM of the evidence generation business Optum Evidence Engine. And prior to UHG, Michael was Chief Strategy Officer of Seven Bridges Genomics, Chief Scientist of Lockheed Martin's Health and Life Sciences Business Unit, and a Lockheed Martin Fellow. He holds a PhD in Biomedical Sciences from UCSF and a BA in Molecular Biology from UCSD. And welcome, Michael. Thanks for leading us in the charge. And anything else you want to say about yourself, feel free to go. It's your show now.
Cool. Thank you. Thanks, Steve. Thanks, everyone, for joining this morning. When Dave asked if I would take on the moderator role, I said yes, of course, because Dave and I go way back. And also, this topic's near and dear to my heart. When I was funding sponsored research, I had a similar type of event that I attended, and I found some great researchers that I was able to sponsor, and we did some prototyping, and that led to some groundbreaking work, I thought, in large-scale data platform development for genomics back in my days at Lockheed. So I'm happy to give back here. The other thing that motivated me to promote this conference was the topics that we're talking about today. I think, as Steve mentioned, in my time at United Health Group, where I had a front row seat onto what payers are considering when it comes to integrating new technologies into precision medicine, there's a number of issues that I think are at play. And one of them is around the challenges of germline hereditary risk being incorporated into clinical development. I think that topic has struggled for close to 20 years, largely because of the clinical utility the difficulty of relating clinical utility of germline hereditary cancer or genetic risk to clinical decision making. And what I really like about the topics today is these researchers are focusing on biomarker development in the genomic space, but closer to disease and closer to disease processes. So I think what you're going to hear today is some research around how epigenetic biomarkers are being discovered and developed and being driven closer to applications, and I think have a more interesting, greater chance of being integrated into clinical decision making. And one of the reasons I say that is the aspect of timing. Hereditary risk has a real difficulty because the timing in which those decisions are affected is very long range. And in our current applications in healthcare, there really isn't a lot of patients for long-term prevention and also long-term risk. I think what's possible here now with more disease-driven biomarkers, I think there's a lot of opportunity to shorten that time window and to have a greater chance of integration. So I know those are more down the road in terms of what the researchers are going to discuss, but I think it's very relevant to showing their ultimate value. And as you consider developing technologies around these topics, is something you might want to consider as well. So let's jump in. I think we have a couple of administrative things we need to talk about. So first is, since we have 3 presenters, Steve, how about we give them 15 minutes or so to present, followed by 10 minutes of Q&A. Do we have a time budget for that?
That's fine. Actually, I forgot to mention at the top that unfortunately one of our researchers who was originally scheduled to participate, Dr. Paul Rothman from UCSB, unfortunately had a family situation he had to attend to and couldn't attend today's. So yeah, we have a little more time in the schedule. If you'd like to expand into that, and if the researchers are okay with spending a little more time with us, we'd love it. Yeah, thank you.
Okay, great. And then as far as questions go, you can put your questions in the chat, or we can I don't know if there's a hand raising function in Zoom. I'm not a good Zoom person, but or we can ask to just have this as a discussion. There's a small group of people on the call today, so if things turn into a discussion after each of the presentations, I'll do my best to moderate that. I think the order of presenters is going to be Dr. Pellegrini. Is that correct? Followed by Wei Li, and then as Paul Boutros comes online, we'll see his presentation. I haven't prepared a bio-type intro for each of the presenters, so if you don't mind giving a little bit of introduction to yourself and your lab the way you would like, and then jump into 15 minutes worth of presentation, and then we'll collect questions in the chat, or we'll have a discussion after each of the presentations. And before we start, I'm going to give Alfredo the best background award so far. I really like that. So
thank you.
So Matteo, do you mind going first with your topic?
Okay, so I was going to give kind of a very high-level view of DNA methylation and why we study DNA methylation and some sort of foundational issues related to DNA methylation. And I think you'll hear of other applications by some of the other speakers today. So again, just my bio to start off. I'm originally a physicist. My PhD was in physics. But then I sort of started working in the biophysics and biology space. And this eventually led to my founding a company, went after my postdoc, and then I moved to Merck Pharmaceuticals for a while, and then finally came back to UCLA in about 2005, so about 20 years ago. And during these past 20 years, my lab has really focused on computational method development for the analysis of both epigenetic and transcriptomic data. So less so on the genetic side, more on this these epigenetic topics. And I'm also a founder of another startup that is going on right now that touches some of the topics that I will cover today. So why DNA methylation? So as many of you may already know, there are 4 bases of DNA, A, C, G, D. And some of these bases get covalently modified, and we focus on one specific covalent modification, which is the addition of a methyl group to cytosine. And within the human genome, this typically occurs at cytosines that are followed by guanines, so CpG dinucleotides. And because of the symmetry of DNA, if you have a CG on one strand, you have a CG on the other strand too, the complementary strand. And so you end up getting 2 methyl groups in the major groove of the DNA. And so these then affect the way the DNA is sensed by all kinds of transcription factors that are binding to DNA. And so it has a profound influence on the way DNA is interpreted in each cell. And so the high level view is that while every cell in your body has the same 4-letter sequence, right, ACGT, these patterns of DNA methylation are dramatically different from cell to cell across your body. And so they enable different cell types to emerge, the phenotypes associated with these cell types, and they also change with time. And I'll talk a little bit about the time aspect as well. So it's a dynamic process. Unlike genetics, which is static, epigenetics is dynamic, it's plastic. And that's why we're interested in studying it. So while we use the same molecule, DNA, we're studying sort of dynamic aspects of DNA. So how do we measure DNA methylation? So there's many techniques out there that can be used to do this. Probably one of the most popular is the use of DNA methylation arrays that Illumina sells. And so you kind of get a little schematic here. You have these beads with probes associated with them, and these bind to bisulfite converted DNA. And so what bisulfite conversion does is it allows us to distinguish cytosine from methylcytosine, because we treat the DNA with this chemical reaction that converts the unmethylated cytosine to thymine, but doesn't convert the methylcytosine. And so by mutating the DNA, we can then distinguish between cytosine and methylcytosine. And once we have the converted DNA, we can detect whether this conversion has occurred by hybridizing this converted DNA onto oligos that are bound to a bead in the case of DNA methylation arrays, or you can sequence the DNA. And so there's lots of techniques associated with bisulfite sequencing. There's also some of these newer single molecule DNA sequencing platforms like PacBio can also detect the difference between cytosine and methylcytosine directly without any bisulfite conversion. And more recently, there's also been some interesting developments in using kind of qPCR-based approaches to detect cytosine from methylcytosine. So it's an active area of research, and there's many technologies that have emerged to carry out the measurements of DNA methylation. In my lab, we've kind of been focusing on one of these approaches, which we call targeted bisulfite sequencing. And so it's shown schematically here. This is probably a bit too busy for this group, but just to walk you through, we typically extract DNA from samples. So often we work with either blood or saliva or buccal swabs, easily accessible samples. We then prepare these DNA fragments into libraries, which are used for sequencing on Illumina sequencers. And so there's multiple steps where you have to ligate adapters. What we then do, because it's expensive to sequence all the DNA within your sample, which we refer to as whole genome bisulfite sequencing, we tend to do targeted sequencing where we design panels of probes that allow us to capture specific regions of the genome that we're interested. So it's kind of the analog of an array, but it's done in solution. We don't have sort of a solid array type platform. And so after we capture the DNA of interest, we then do this bisulfite conversion, again, to distinguish cytosine from methylcytosine. And then this is sequenced. And this allows us to generate large data sets with hundreds of samples, at fairly low cost for less than $100 per sample. So we can start to study the association of DNA methylation patterns with all kinds of other metadata that we can collect about these samples. And then in terms of, what determines, so I said DNA methylation is plastic, so it's different from cell to cell, it's different across time. And so what determines these differences in DNA methylation? And one simple way to think about it is it's basically DNA methylation patterns respond to the state of the cell. And so, as you know, each type of cell is expressing a subset of all the possible genes that we have in the genome. And this is mediated through modifications of chromatin. And so, for instance, some promoters can be active in certain cells and repressed in other cells. And these modifications of chromatin and promoters then directly interact with the enzymes that are responsible for methylating DNA. And so we have, a very deep mechanistic understanding of how DNA methylation is targeted, how it changes from cell to cell, and how it's associated with the cellular state. So it's really a readout of cell state that's telling us essentially for these particular types of cells which genes are being expressed and which genes are not being expressed. So if that's the case, then why focus on DNA methylation? So we could look at gene expression directly, right? And so there's many assays that look at, instead of DNA, they look at RNA to measure gene expression. You could look at chromatin or histone modifications. But instead we focus on DNA methylation. And so why? And so one way to think about it is, there's lots of timescales within genomics, right? We think of genomics as having lots of different data modalities, but these different data modalities are associated with different kinetics, different timescales. So obviously, if you're interested in genetics, you're looking at invariant processes, right? So the genetics is not going to change throughout your lifetime, except for, rare examples of somatic mutations or tumor-induced mutations. DNA methylation is not obviously invariant, but it is quite stable. So what distinguishes DNA methylation from the other types of genomic modalities is the fact that it changes very slowly. So in order to influence DNA methylation, you need some kind of process that persists over weeks or months, and generally, slowly changes the patterns of DNA methylation. And this is in contrast to the other modalities where you're looking at gene expression. We know gene expression is constantly responding to the environment every time you eat food, every time you go to sleep. Gene expression is cycling throughout the day. It's a very, very dynamic process. So this, again, when you're thinking about the different types of data that can be collected with genomics, it's important to think about, what type of processes you're trying to capture, whether they're short, immediate response processes, acute processes, or whether they're kind of slower, chronic processes. So DNA methylation, apart from its time scale, is also influenced by, lots of factors, right? And so one of the things we do in my lab is we try to understand what all these factors are that drive DNA methylation patterns. And so one of the most important components is the fact that, as we said, different types of cells in your body have different methylation patterns. So if you gather a sample and measure its DNA methylation,
I think we may have lost Matteo for a minute. Or we lost me.
I thought it was me.
Yeah.
Am I back?
Yeah, I
You
think
did let
we
me-
lost Matteo.
Uh-oh. And he's frozen.
Well,
So while he's coming back, I really appreciate the point he's making about timescales. And one of the questions I was going to ask, and hopefully we'll talk about, is sort of the labile nature of biomarkers. And is Matteo back?
oh, sorry, did I lose you?
Yeah, you froze for a while after you were talking about your timescale slide, and then you went on to what your lab was doing. And I just wanted to also insert a time check. So you have about 6 minutes left.
Okay. Yeah, so I was, I think, at this slide, which was showing all the different factors that can influence DNA methylation. So as I mentioned, different cell types, that the heterogeneity of a sample can have a big effect on DNA methylation. I'll talk a little bit about how aging affects DNA methylation, but then there's other factors, physiological factors, genetic factors, disease factors. So part of our research is to try to understand how all these factors influence DNA methylation. So at the aging level, that we've known for now over a decade that there are very slow processes that impact DNA methylation over the course of our lifespan. So this slide just shows one single CpG dinucleotide in the genome, and its methylation levels, which are usually measured between 0 and 1, so 0 being unmethylated and 1 being fully methylated. And this is the lifespan over, population going from birth at 0 to centenarians. And you can see that for a single site in the genome, we have this kind of perfect tracking of age with methylation. And so this is kind of a remarkable process, because there's not too many processes in biology that occur over such a slow time scale, right? Typically, when we think of biology, we think of minutes, days, weeks, but we rarely think of a century. But within this DNA methylation paradigm, we can really track a century-long process just by looking at the methylation of a single site. And so this has led to the development of things called epigenetic clocks that can essentially predict the age of an individual based on their DNA methylation patterns. And there's dozens of companies that are now selling these products, where you send in DNA either through blood or swab or saliva, and they will tell you what your age is. And they can do it quite accurately, typically, within 4 or 5 years. Now, in the case of humans, we know our age, so there's not so much interest in knowing what our age is. But the promise is that there's a difference between the predicted epigenetic age and the actual age, and that delta is measuring something about our aging rate. So people who have faster epigenetic aging than their actual age are presumably aging more rapidly. whereas people who have slower are aging more slowly. And so that's part of the goal of this field, is to use these kind of biomarkers to lead to slower epigenetic aging and hopefully better health outcomes. Now, how do we know that epigenetic clocks track environment and not just, our actual age? So we've been doing experiments with other animals where we can have, dramatic perturbations. In this case, we looked at marmots, and marmots spend a large part of their life hibernating during the winter. And so we asked if this epigenetic aging clock is impacted by hibernation, right? You might expect that while the marmots are active during the spring and summer, they're aging rapidly, whereas while they're hibernating at low temperatures, they're aging slowly at the epigenetic level. And that's in fact exactly what we find. So there's this seasonal pattern of epigenetic aging that perfectly tracks their epigenetic aging. And so this shows that the rate of epigenetic aging is not fixed. It's not just measuring time, but is influenced by environment. And if you can slow metabolism dramatically, like you can in marmots, then you can affect this epigenetic aging. And another example, we've looked at dogs, for instance. So the nice thing about dogs is they have, huge physical differences across breeds, right? We have very small dogs that weigh maybe, less than 10 pounds to dogs that weigh over 100 pounds. So these kind of differences cannot be studied in humans because we don't have such dramatic differences in body size. But so if we track this epigenetic aging across dogs, we find that in fact that the small dogs are aging epigenetically much more slowly than the large dogs, which fits with our understanding and our data that shows that also, large dogs tend to age more rapidly and live less, have sort of shorter lifespans than small dogs. So again, this reinforces the notion that this epigenetic age is not just tracking actual age, but it's tracking some molecular correlate of age that has health and disease implications. And as I said, I mean, DNA methylation is not just influenced by age, it's influenced by lots of facts. So we see very dramatic differences in epigenetic age between males and females on the sex chromosomes. And so we can use epigenetic information to perfectly track the sex of individuals, and also try to identify interesting cases like hermaphrodite individuals or individuals that have ambiguous sex. What happens to their epigenetic sex is interesting. And we also found that in dogs, again, where we were able to study the effect of spaying, that there was a dramatic impact also on the methylation. And so we could predict whether dogs had been spayed or not based on their methylation patterns. And the last example I'll give you is kind of a very different example, which is from the field of human diagnostics. And this is obviously a very large field, and I think you'll hear more about this today. But the idea is that we want to be able to track the presence of tumors within individuals through the DNA. And so the way we do that is that tumors can shed DNA. Every cell in our body has some kind of half-life, and so eventually these cells die and they release DNA into the plasma. And so obviously the DNA from all these cells is the sequence of DNA is very similar, but the methylation patterns, as I mentioned, can be very cell type specific. So if we measure the DNA methylation patterns through this bisulfite sequencing of the DNA in the plasma, we can then try to understand what type of cells release that DNA. And so here in this example on the right, we show that if we collect plasma from the blood of patients with or without cancer, and we look at their bisulfite sequencing, and we just kind of, in this case, take all the methylation patterns and project them into 2 dimensions using these dimensionality reduction techniques, we can get very good separation between individuals that have, have no disease versus people who are known to have cancer. And so this is the promise of these liquid biopsies that's being pursued by many companies, and we're doing a little bit of this work in my lab as well. And so this is, again, is a very exciting application of DNA methylation that could be used for early diagnosis of disease, not just cancer, but we're also applying this to neurodegenerative diseases have shown that we can detect diseases like ALS by looking at methylation patterns in the plasma DNA. So just in conclusion, then, hopefully I've convinced you that DNA methylation is this interesting biomarker. It's impacted by many factors such as age, sex, cell composition, and disease. It can be measured very well. There's many techniques for doing it, but it can be measured cost effectively and accurately. And so it lends itself as a biomarker. I've given you the example where you can use this DNA methylation to track age and also to understand how various environmental or physiological factors can impact the rate of epigenetic aging. And lastly, there's also promise for human disease diagnosis as well. So I'll stop there.
Thank you, Matteo. That was a great introduction to epigenetics based on methylation patterns, and great work in your lab. You also mentioned in our discussions that your lab has done some pioneering work on tools and techniques that make detection of these biomarkers more tractable. Anything you want to say on that in terms of like commercial applications or potential collaborations with your lab?
Yeah, so the technique we've been developing, I had that one slide on the targeted bisulfite sequencing. So, there's always a balance between, cost and the number of regions you measure, right? So at one level, you can always measure the entire epigenome by doing whole genome bisulfite sequencing. But that's still, even with, the reduced sequencing costs that we see every few years, it's still fairly expensive. So, it's tractable for certain projects, but not for every project. And so what we've tried to do is combine the rapidly, decreasing costs of sequencing with our ability to also target that sequencing to the regions of interest. And so that's what we've developed. And the other point that I wanted to make that I didn't make in the presentation is when you're doing sequencing, there's coverage is important. I mean, the number of regions you measure is important, but also the number of reads that you obtain per region, because in order to obtain accurate estimates of methylation, you really have to sequence the DNA quite deeply. And so these targeted methods allow us to find kind of the sweet spot where we have high coverage at each region, so we can get very accurate methylation estimates, and also enough regions that we can sample the parts of the genome that we think are impacted by the particular factor that we're trying to study.
Awesome, thank you. Any questions from the audience?
Matteo, I have a question regarding, that bisulfite use in cancer detection. And I really thought that there is a good opportunity about applying it in the real world scenario, because you go to the doctor and then you get screened for multiple cancers at different times, versus if you have one test that can give you an idea that you need to get the screening done using bisulfite, that could be more useful. But you don't want to raise false scares unless you have, a directed approach that can tell you. So have you thought about that in developing that further?
Yeah, so one of the promises of using the plasma DNA methylation as a cancer diagnostic is that, well, A, it's non-invasive, B, it can potentially capture cancer early on, before it, has become, while it's potentially all still treatable. And also it can differentiate between different types of tumors. So, because it's trying to detect the origin of the DNA, different types of cancers or cancers that originate from different tissues and cell types will have slightly different methylation patterns. And so that's the other promise, is it's not just detecting cancer in a generic sense, but is detecting a type of cancer. So, this is a huge effort in industry right now. There are companies like Grail that are already selling these types of tests, and they claim that, with fairly high accuracy, they can not only detect the cancer at early stage, but also what type of cancer it is. So you can then go in and screen. There obviously are going to be false positives, and that's the big challenge here. If you use these technologies for screening, what happens to the people who get, false, cancer positive results? There'll be obviously anxiety and all kinds of things. So that's a challenge the field has to deal with. But it also promises to save a lot of lives by catching cancer earlier than the current screening methods do.
And plus, the real issue here is that people go to the doctor for annual checkup and they anyway give blood for other purposes, but they still don't know what's going on, with their body, unless if there is a quick test that can be done and ask you to do further checks if it's reliable.
Yeah, I think that's the vision, that as these tests become more accurate and more affordable, they'll be used quite broadly, right? So there's no reason. I mean, there's already DNA-based, yearly cancer screens for like colon cancer and other types. So the idea is this would be kind of a pan cancer screen that could detect, dozens of different types of cancers.
Okay, thank you.
Thank you for that question. There's a question from the chat too, Matteo. What do you think about nanopore type sequencing for detection of methylation? Does that help in terms of the accuracy or the cost or throughput? What are your thoughts on that?
Yeah, so as I mentioned, with some of these single molecule, either nanopore or PacBio, you can detect the fifth base, this methylcytosine, directly without doing any conversion. And so those are very powerful and potentially promising ways of doing it. The one thing you can't yet do very accurately with either nanopore or PacBio is targeting. So they're useful if you have large amounts of DNA, and you don't want to do targeting. If you do want to do this kind of targeted approach where you're only sequencing a small fraction of the genome, then those technologies are not particularly effective for that purpose. And that's why the vast majority of the field is still relying on these kind of bisulfite conversion based assays combined with short read sequencers. But inevitably this is going to change, and both nanopore and PacBio will likely improve their ability to target their sequencing to various regions. And then there's also the issue of input, right? It depends what you're studying. If you're studying blood and saliva, it's not a problem. But if you're studying plasma DNA, there's minute amounts of plasma DNA. And so some of these assays as well would require amplification. And in that case, you lose the methylation information if you start amplifying. So they're not necessarily yet usable for those kind of applications with very low input.
Got it. I didn't appreciate the targeting aspect of nanopore. So that's super helpful for me too. Any other questions from the audience?
Just a quick question. As a consumer, I guess that's what I would consider myself. What's your recommendation, Matteo? Do I want to look for things that are FDA approved, or are these tests, they don't have to go through that kind of a rigor? What's your thought on that?
So it depends. If you're talking about the epigenetic aging test, there's no FDA involvement. If you're talking about the cancer, obviously there is. Yeah, so there's this whole kind of booming space of wellness, right? That's not medical, not disease diagnosing. It's just kind of trying to assess whether you're aging well or not aging well. In that case, the FDA tends not to get involved, right? As long as there's no specific disease. So, it's a booming market. There's lots of companies selling these. I think we're very early stages, and I think, it's more at this point, it's a little bit more of an entertainment product than a medical product. But people are fascinated. People do 23andMe, people do all kinds of things. They just like to know a bit more about themselves. And even if the science behind it is not completely nailed down, it's still interesting to play around with these things as they're evolving and see. And again, they can also be applied to animals. So you don't. There's obviously we're actually thinking of using them for animals. Like, for instance, we're predicting the age of dogs or other animals where other techniques for estimating age are not nearly as accurate. So it's not just in the human realm.
So can I dig into that one a little bit deeper? So the idea of having an epigenetic marker related to aging, if you consider aging a disease process, what are your thoughts on using these types of biomarkers for clinical development or preclinical development, looking for interventions into the aging process?
Yeah, so that's a big space. So that was one of the promises of these epigenetic clocks, especially in the human setting, is that you could use them as biomarkers for efficacy of aging treatments. And so a lot of the companies that are trying to develop, anti-aging compounds will often use these epigenetic clocks to see if these compounds are having an effect. And there have been studies, still early days, but there are studies that show that some kind of dietary or intervention with various compounds can lead to changes in these epigenetic clocks. So they are, as I've shown you in these examples, they are malleable, right? They're not fixed. So you can intervene with either diet, lifestyle, or drugs and slow down the rate of these epigenetic clocks. So I think that's a huge area in a huge application, especially in therapeutics.
Thank you. All right, we're out of time for the presentation and Q&A. Thanks, Matteo. That was a great introduction to our topic on epigenetics. And if you want to follow up with any collaborations or discussions with Matteo, you can get his contact information through Steve or through the invite. Our next presenter is Dr. Wei Li from UCI. I'm going to turn it over to you, Dr. Li. And I'll give you 15 minutes for presentation, and then we'll do 10 minutes of Q&A. And I think you've been set up for your topic quite well, I think.
Oh, thank you. Thank you for the opportunity. Let me try to share my screen. But somehow my Zoom doesn't work very well. Okay, here we go. And so my name is Wei. A little bit about my background. So I've been working on genomics, computational biology, and epigenetics for more than 25 years. That means I'm quite old right now. So I was one of the founding members of the computational biology department in BGI, the world's largest genome center in China. And my first paper as a graduate student was the 2001 Nature Human Genome Sequencing paper with Eric Lander as the first author. Then I came to the States to do my postdoc training at the Dana-Farber Cancer Institute in Harvard Medical School. Then my first faculty job was in Baylor College of Medicine for 12 years before I moved to UC Irvine as an endowed chair and a professor of bioinformatics to lead a new division of computational biomedicine. Can you guys see my screen okay?
I can now, yeah, thank you.
Okay, good. So really, my topic today is multiple modal epigenetic sequencing analysis for cancer prognosis and early detection of brain disorders. So by the way, I have a conflict interest disclosure. I'm the board member and scientific advisor for two startup companies, Children Med and Helio Genomics, for multiple years. And I believe those two companies are ready for IPO, hopefully next year. So let me go to the next one. So everybody know liquid biopsy is really the holy grail in diagnostic medicine, as Matteo mentioned. And a lot of companies are focused on DNA methylation on cancer early detection. And so the whole idea is to get a blood draw from patients followed with some kind of molecular measurements and to detect the diseased cells in human body. But we really focus on the epigenome of the cell-free DNA human plasma, which the cell-free DNA are predominantly nucleosome protected DNA from the dead cells. And in general, people believe, for example, for cancer patients, 1% of cell-free DNA are derived from the tumors, from the tumor tissue, and 5 to 10% from your normal organ, lung and liver, and more than 80% are derived from your immune cells, right? That's really the big. So basically, cell-free DNA is a mixture of all the dead cells in your human body. So this is super important because cell-free DNA epigenetic signature really represents the both diseased cells and the immune reactions, right? So just as a seeds in different soils produce fluids with very different flavors. Identical diseased cells, for example, the seeds, paired with diverse immune response, the soil, might result in very different clinical outcomes, right? That's really our hypothesis we are testing, but we have very strong preliminary data to show that's indeed the case. And the often overlooked immune responses, the soil can really enable liquid biopsy for broad spectrum of disease beyond just early cancer detection, right? So we have a very unique technology. We call it MESA, Multimodal Epigenetic Sequence Analysis of Cell-Free DNA. And we work with a twist to do this target EM-seq with a twist methylome. So EM-seq is enzyme conversion of the DNA, very similar to bisulfite conversion, but it's instead of chemical conversion, this is enzymatic conversion. So it's very mild and it's perfect for clinical samples. So we can routinely generate 250-fold very deep sequencing on the entire methylome of the human genome, which includes 4 million CpGs and 130 megabase regions in very tiny plasma for very reasonable cost. Right now, the reagent cost is roughly $400. So it's very affordable. And in my opinion, there's really no reason to design your customer panel anymore because the really genome-wide methylome targeted sequencing is really affordable now. Then from the data, we can really infer, we can computationally infer methylation information, which was the original purpose of the methylation sequencing, also the nucleosome organization. Because as I mentioned before, the cell-free DNA are really the nucleosome DNA in the plasma and the nucleosome protected DNA in the plasma. And in the paper we showed, because nucleosome and the DNA methylation are two very distinct and highly complementary epigenetic mechanisms for gene regulation and for the disease risk. So by combining the multiple modalities together, we can really achieve a much better disease detection accuracy, right? So this is the data we generated with a twist methylome target EM-seq. And in addition to methylation, when you look at the reads pi up, basically the density of the reads, you can very clearly see this roller coaster pattern, which represents the nucleosome in the genome. And we can observe the nucleosome free in the promoter and the end of the transcription. Then by comparing cancer with the non-cancer, we can very clearly see in this case, the gain of nucleosome, the loss of nucleosome in cancer in gene 1 and the gain of nucleosome in gene 2, so that we can use the nucleosome gain and loss as a distinct epigenetic modality and the future for cancer early detection. And we also have very strong track record in successfully commercializing liquid biopsy test. Right now, I serve as a chief bioinformatics advisor for Genomics, which is a startup company located in Irvine, California. And together we developed the Helio liver test, which we believe is the first methylation test case for liver cancer targeting the high-risk individuals with liver conditions such as liver cirrhosis, fat liver disease. And this test has now been approved by China FDA in 2023. And this test is LDT in the US now, and we aim for US FDA submission in early 2024. So for the pipelines in my lab, the first pipeline is really for the prognosis of cancer for better treatment decisions. The unique resource we have is roughly 3,000 breast cancer samples with more than 20 years of follow-up. So this 20-year follow-up is really the key to distinguish long-term survivals versus short-term survivals. So in the market, there are several kits for breast cancer prognosis, such as Oncotype DX and the MammaPrint. But those tests rely on gene expression from tumor samples. So they are not liquid biopsy, and they only look at the seeds, and they ignore the immune reaction component. And they don't have long-term data past 10 years. So in the figure, I show you some of our preliminary data from our 20-year follow-up collections. We can clearly see on the left figure, there's a very dramatic methylation difference between long-term survival and short-term survivals. And remember, those are plasma we collected more than 20 years ago. And because of the beauty of DNA methylation epigenetics, those samples remain in high quality for epigenetic sequencing. And the other cancer we are working on, including prostate cancer, with 10 years follow-up, we got the blood at the time of surgery for primary cancer removal surgery. And with 10-year follow-up, we know which patients can survive more than 10 years, which patients has recurrence within 1 or 2 years. So the second big direction is really brain disorders for early detection. So sorry, it's really for brain disorder early detection. The goal is to enable clinical trials targeting early onset patients. And the hypothesis is different brain disorders will induce very different immune responses, and which can be detected through our MESA multiple modal epigenetic sequence analysis. And again, we have a very unique resource. We have roughly 2,000 plasma samples from ALS, which is a very deadly neurodegenerative disease. And this disease currently has no laboratory or imaging parameter that allows the detection of ALS, and the lack of biomarker and test really greatly delays the diagnosis of ALS and really delays the enrollment in clinical trial. As we believe that's part of the reason there was no drug being successfully developed for ALS, because there's no way for those farm companies to recruit early onset patients, right? Once they recruit ALS patients, it's already in the very late stage. The damage was already there. It's very difficult to reverse damage. So that's why the drug development is so difficult. But if they have a drug to prevent the progression of ALS, then they will really have the need to recruit early onset patients to test the efficacy of the progression reduction. So the last pipeline is really MESA liquid biopsy for suicide prevention. We have no commercial plan for this particular test. We are doing this purely for our scientific interest. And right now, there's no validated biomarkers that can be used clinically to assess or estimate suicide risk. And we all know the major depression disorder is the most common diagnosis in individuals who died by suicide, but only a very small portion of depression patients are at risk of suicide. And really the question is why 5% of major depression disorders committed suicide, while the remaining never perform suicide, right? So our hypothesis is there's a strong molecular basis for MDD suicide. And indeed, our preliminary data showed the hyperactivation of stress response genes in their immune response are really a hallmark for the suiciders. So again, through the psychiatric clinic at the UC Irvine, we have 200 MDD suicide versus 200 MDD non-suicide with 5 or 10-year follow-up. And so we think we can really develop a new epigenetic signature to predict the suicide risk molecularly for those major depression patients. Then the intervention is so straightforward, right? The clinician can double the amount of depression drugs, right? And they can really watch the patient more closely and inform their family members or friends to watch the patient more closely to prevent suicide, hopefully in the future. So as a summary, we have a new technology called MESA to enable liquid biopsy for cancer prognosis and early detection of brain disorder. I talk about the breast cancer project, ARS project, right? For the breast cancer, it's really for the better treatment decisions. And but for ARS, the goal is to enable the clinical trials targeting early onset patients. And the last one is really suicide prevention. So how many additional minutes do I have?
1 minute.
Okay, cool. For 1 minute, let me show you another project we're working on, which is super cool. We recently developed this super cool tandem repeat genome aggregation database, collect the data from roughly 340,000 whole genome sequencing from healthy individuals. And for each individual, we can really genotype their tandem repeat expansion. For example, 3 copies in people number 1 and 6 copy in people number 6. And the tandem repeat expansion has been indicated in many neurodegenerative disorders, such as Huntington disease, Fragile X syndrome, but their role in other diseases are completely unknown, right? So based on our database, we can really identify, because this is really a background database for tandem repeat expansion across the entire human population. Based on this background, we can really perform the outlier detection for particular tandem repeat expansion in patients, right? So those outlier tandem repeat expansion is very likely to be pathogenic or causal for that disease. For example, through this database, we can readily detect the Huntington disease associated tandem repeats and the C9 operating frame associated tandem repeats expansion in dementia and motor neuron disease. And really the next step is to do the TR-based genome-wide association studies for all the human disease and the outlier TR detection for the diagnosis of rare disease, working with the GREC consortium, which is a genomic research to elucidate the genetics of rare disease. Okay, so that's all I can share with you today. Thank you so much for your attention.
Thanks, Wei. That was terrific. So there you go. More on taking methylation signatures and applying them to early detection of a variety of diseases and conditions. I have a bunch of questions, but I want to open it up to the audience for your questions. Any?
I have a question.
Yeah.
So, there are companies like Prestige Bio that have got quite a lot of traction. They just raised $57 million. Their technology is basically very similar to what you're describing. How do you differentiate yourself from existing liquid biopsy technologies that are revolving around the nucleosome and the methylation? And that was going to be my question for the previous presenter as well.
So I would say from the sequencing technology perspective, there are really multiple similar technologies available, right? The PacBio you mentioned, the nanopore, and people combine methylation with different modalities already. But what's unique about our cohort of our pipeline is really the large collection of patient material and our machine learning expertise that can really combine different modalities to develop a very unique epigenetic signature for the disease prognosis and brain disorder early detection. Because we try to stay away from the cancer early detection sector, which is super busy right now, as you all are already aware. And really my goal is to develop a liquid biopsy for other unmet clinical needs that was not the focus of those big guys.
I have a comment on your question too. So sitting at the payer level, we got a lot of inbound on people wanting reimbursement for liquid biopsy tests. And there's a variety of technologies, and as we're learning, there's a variety of applications, and they're all very well founded in science. The gap, in my opinion, is validation of those technologies toward clinical endpoints.
Right.
That's the gap for multiple cancer early detection. It's the gap for liquid biopsy for lung cancer still. The Helio test is going through its validations and right now. And I think the foundational work on clinical validation that Dr. Li is doing in his lab, and the way he describes it, he's got samples with multiple years of clinical follow-up, I think is the way to start and to build differentiation around the evidence you have for your analytical validation and your clinical validation. I think that's where the differentiation is going to exist in these tests as they go to market. And I would I'd turn this comment into a question for Dr. Li to ask him to tell us more about your evidence generation plans for the samples that you have and how you see developing these applications toward clinical. And I know you have experience in this area, so I don't think it's a hardball question.
Well, fundamentally, I'm a basic scientist. I do a lot of research purely based on my own personal interest. And sometimes we didn't think enough about commercialization, which is good or bad, right? So really my goal is to take advantage of those, very prestigious sample collections that are available to me and develop epigenetic signatures to enable the prognosis and early detection. Then at that point, I think my plan is to either work with a clinician in academic setting to do the investigator-initiated clinical trials, or if I can find an investor or commercialization partner, we can form a commercialization entity, right, to do the commercial clinical trials for those tests developed in the lab. So that's really the plans, and I'm open to every possibilities. For example, for suicide prevention, I know right now there's really no clear plan for insurance reimbursement, but I don't really care that much. I think if our test really works, can save lives, that's the most important part.
I would add that if you want to see these tests to see the clinical application, reimbursement is one thing, but also getting provider adoption. And in order to do that, you need the same kind of evidence for clinical validation for the test and ultimately clinical utility, so they use it. And in our discussions, you and I talked about, the potential to piggyback this test onto something like GeneSight that's used to look at, a pharmacogenetic picture of major depressive disorder. And in that case, the blood draw is already there. If you had a clinically valid test that said on top of that result, you could say that there is a risk of suicide for a particular patient, that's got short-term clinical application. And it takes me back to my theme in the introduction about how these biomarkers are, I think, very relevant because they impact short-term clinical decision-making. Now, you still need to validate the technology and produce that analytical validation. And the clinical validation that you're doing in the lab and with the samples that you have is a proper step. And then, as you mentioned, the third step is to do the prospective, more structured clinical development around these tests. I think that's the necessary flow.
Yeah, but that's super expensive, right? So that's why I'm super interested in working with the commercial partners to pursue those directions.
That's exactly why we're here. Any other questions from the audience?
Yeah, I have a couple of comments. Great presentation, both of them. So my focus is oncology, but I'm really impressed by the psychological testings done. I think funding would be great for funded studies. But going back to your cancer progression prediction, we lack information in terms of liquid biopsy in GBM or brain mets. any plan on doing studies on that? I know you know, institutions like ourselves would be interested in doing studies in terms of, like, there are studies done on breast and prostate and all the solid tumors, but brain mets, GBMs are the ones that are less known because of the nature of liquid biopsy. Any suggestions? And there was another lab that we would send our CSF data to them, but any you know, high yield in that area?
Yeah, that's a very good question. Actually, my former postdoc, who is a faculty member right now, really published the first paper showing the CFS demethylation for the pediatric brain tumor detection in Science many years ago. We are very interested in brain tumor for sure. I have a physician scientist trainee in my lab who is a very well-trained brain tumor surgeon. So we have collaboration together to collect plasma samples and CSF from GBM and all kinds of brain tumors, and to enable liquid biopsy from blood for brain tumor early detection. But that's still ongoing. We don't have any data yet. But of course, we welcome any collaborations. So right now, the technology, in my opinion, is kind of mature. The cost is reasonable. The machine learning technology in my lab is kind of ready. So the limitation is, number one, the collection of the samples, and number two, clinical validations, right? As long as you have the sample, either plasma or CSF, we are ready to collaborate and to help you.
Yeah, maybe we can talk later to see how we can collaborate.
Yeah, absolutely.
Thank you.
Any other questions? Okay, I think we're moving on time. Thanks, Dr. Li. We're gonna go to our next presenter, Dr. Paul Boutros, UCLA. Paul, are you on the line?
I am. Can you hear me?
Are. Yes.
Excellent.
Thank you for joining. And appreciate you making the time.
I am just sharing screen.
So you missed the intro. We have 3 presenters, so we're giving 15 minutes of presentation time, followed by 10 minutes of Q&A, and I think we have just enough time for that.
Perfect. Great. Can you see my screen okay?
I can.
Excellent. So I'm going to talk about 2 related things that are happening within my research program, really all focused on, consistently with the last 2 talks, early cancers and improving management of early cancer. But they're going to be different. The first thing I'll do is give a very quick overview of prostate cancer to really nail down the problem that we're tackling here. My team does research in several other cancer types: pancreatic, breast, thyroid, sarcoma, meningiomas, glioblastomas, but I'm going to show the prostate cancer story to be really consistent. Then we're going to go through how we might think about the germline genetics and the clinical applicability there. And then we're going to talk about exercise and how we might think about the utility of genomics and exercise. So let's kind of imagine a prostate cancer patient. What happens? A man at age 50, 60 will go to their family doctor and they will probably get a PSA test. Recommendations here are controversial, a mess, conflicting, and inconsistent. Nevertheless, there are about 30 million diagnostic PSA tests done in the US for men between the ages of 50 and 60 every year. So it is commonly done. At that stage, well, the prostate doesn't actually image very well. Almost no modality is able to see tumors in the prostate accurately. That's very rapidly changing, and hopefully in 2 or 3 years, I'll be able to say differently about that. But instead, physicians will often, but not always, do a digital rectal exam. Basically, they insert a lubricated finger into a man's anus, palpate the prostate, and try to figure out, is there a bump? And you can see these little stick figure men. Over in each case, we've got the blue healthy, the gray who have a cancer, but it really doesn't need to be treated, and the one red individual who has a lethal cancer that urgently needs treatment. And you can see after a digital rectal exam, fully half the men don't have a cancer at all, and 5/6, about 85%, don't have a cancer that needs treatment. At this stage, they might get imaging, MRI, usually a $2,000-ish test, which does a very good job of removing healthy tumors, individuals without a tumor, but is not completely accurate at that and does not fully distinguish aggressive from non-aggressive tumors. At that stage, men get a biopsy. A biopsy is almost always taken through the rectum, and it has a 3% chance of sending a man to the emergency room with a bacterial infection. As a result, men are prescribed antibiotics at the time of biopsy, and prostate cancer biopsy is on track to become the single most frequent medical procedure in the next 20 years. That almost always removes individuals who don't have prostate cancer, and then there may or may not be a DNA test done on the extracted tumor tissue. Again, very conflicting guidelines. And then at that stage, treatment decisions have to be made, with about 50% of men who are treated for prostate cancer being overtreated, receiving a test that they don't need. That's the journey for a prostate cancer patient, from going to the family doctor's office to making a decision of how I'm going to be managed collaboratively with their physician and loved ones. So the value proposition here is really clear. Prostate cancer is the second leading cause of cancer deaths in men. There are literally tens of millions of screening tests done in North America and hundreds of millions done around the world. And we've tried to identify ways to reduce the cost of unnecessary biopsies, the high false positive rate of PSA, and figure out ways to give rapid, cheap information to men to improve these decisions. When I say we, this is the multidisciplinary team. It's myself. I am the director of cancer data science here at UCLA and a faculty member in human genetics. My partners in crime, Rob Reiter, is an MD MBA urologist, prostate cancer surgeon, and Amar Kishan, who is a radiation oncologist, just got promoted to full professor, actually. And so the three of us have been working on prostate cancer in some way, shape, or form for literally decades, and have been focusing on this issue from multiple angles. And so the market opportunity is massive. Ultimately, you can kind of think about two key points. Patients and clinicians want additional clarity for information to make good decisions. And it's increasingly known that prostate cancer has a strong family history, a strong genetic component. And that's been true all along, but people didn't recognize it because the population wasn't as old. And aging is making it more and more evident. Hey, I have 3 uncles who had prostate cancer. I have a father and a cousin and a grandfather. And that now creates a better appreciation of the potential role for genetics. And so we started off focus on germline genetic biomarkers to improve those decisions. Why study the germline in prostate cancer? There's a bunch of really obvious reasons. There's lots of known germline risk factors, but I'll give a couple of other really natural ones. If we think about a typical cancer, in the 3 billion bases of its genome, it'll have 3,000 to 30,000 mutations. Any two of us on this call, assuming we're not first-degree relatives, differ by at least 3 million germline SNPs. So there's massively more germline variability than there is baseline. Another way of saying this is people are far more different from one another than cancers are different from one another. Cancers are actually quite similar in that context. And that's true even if you think about structural variants. The typical prostate cancer will have 1 to 5% structural variance, and the typical germline genome will be 1 to 2%. Very comparable, even for those big picture gains and losses we think as being idiosyncratic to cancer. And so some baseline knowledge from the literature in prostate cancer. 1 in every 25 patients is a BRCA2 carrier, and probably much higher than that in the group that metastasize and lead to lethal cancers. Similarly, common germline SNPs in the population have an odds ratio of 5. So individuals with those are 5 times more likely than the median men to have prostate cancer, which means you could potentially be avoiding 20% of biopsies just by using existing genetic information. And maybe the most key point here is if we take away melanoma and leukemias, which have a lot of interesting characteristics, prostate is the most heritable solid cancer type. So almost 55% of the likelihood of anybody getting prostate cancer is familial. It's just very strongly genetic. You compare that to colorectal cancer at 15 to 20%, or breast cancer at 25 to 30%. Prostate cancer is really an outlier where genetics gives a lot of information. So what did we do? The first thing that we did is ask, can we figure out how germline features might interplay with the lives that people live? And so we took those germline features in the genome that are known to influence the risk of a prostate cancer diagnosis. Those are laid out over here as the columns. And then we asked, did they interplay? Did they affect the methylation status of specific regions of the prostate cancer genome? And every time you see a black background with a red or blue dot, there is one of those associations. We did that in about 500 patients, and then we went ahead and validated them in about another 500. And you can see the discovery and validation are very tightly correlated. So this is a general feature. Germline features are influencing what's going to happen in the tumor. And therefore, maybe not surprisingly, you can find individual germline variants, which are pretty good predictors of patient survival. Here's one example. It's in a gene that's associated with hormone synthesis. And you can see individuals with a certain allele of it have 90% 10-year survival, whereas those who are of the poor outcome variant have about 60% 10-year relapse-free survival. Great. So germline SNPs are giving us some clinically useful information. Well, what does it tell us about how the tumor is actually evolving? We looked at about 650 tumors. We did everything you could like to these DNA, RNA, protein sequencing, and we asked how the germline is influencing the tumor evolution. And here's the kind of summary plot of this. Each of these rows is an individual gene that's influencing cancer evolution. Some of these are very clearly druggable. Some of them, there are no way to drug. And every time you see a black background or gray background with a red or blue dot, that means that what is happening in the tumor is influenced by the germline genetics. How much influence? Well, 4 times. So we can say, for example, for this mutation, that the man who has this germline SNP is 4 times more likely to have that mutation than the man without it. So what does that really tell us? Well, it means that at birth, we can predict what is going to happen in a man's prostate cancer 70 years later. The ultimate early prediction. These are not perfect predictions, right? You can see it's a 70% accurate prediction. But here we're saying, based only on the germline genome, what is going to happen in this tumor. And we make a pretty good prediction. It's not perfect, but it's easily sufficient to guide other test decisions and to influence the way we think about allocating other tests. And of course, this is only one example. We've been able to do it for all of the tests that I showed you over here. So in fact, just to give you a sense for it, these explain 30% of the ancestry differences that happen in prostate cancer. And so what we've put together is a very tight targeted sequencing panel linked to some really interesting technologies for library preparation and automation. It looks at about 0.1 or 0.2% of the genome and allows you to, for about $40 or $50, measure everything that you could care about in prostate cancer. The entire germline genome, BRCA, all the features that might be interesting there. A couple of reasons why we might include something like BRCA, which of course is measured in other ways, one of which is it allows us to have an immediate clinical actionability because BRCA testing is clinically indicated for metastatic and high-risk localized prostate cancers. We've now accrued about 12,000 patients across different cohorts with outcomes ranging between 5 and 25 years, which we are now going to do this retrospective validation to demonstrate the utility of this on primary endpoints, which would be what we aim to have as a package of evidence to support guideline changes, which we then hope would be key in driving commercializability and actually just helping patients. Okay, I have like 3 minutes left. Let me quickly talk to you about, that's all great, but all I told you about was diagnosis. What can we actually do to help patients? Well, really, the only thing you can do to help patients in prostate cancer is exercise or cut out their prostate. There are not other options. So yeah, what does exercise do and why is that interesting for this audience? We've put together a really exciting clinical trial of exercise, investigator-initiated academic. We, as Lee Jones at Memorial Sloan Kettering and myself at UCLA, we bought a fleet of 50 treadmills. We bought watches, iPads, and we shipped the treadmill, watch, and iPad to patients who have just been diagnosed with cancer. And when I say just been diagnosed, within 3 days of diagnosis, they get the treadmill, they put on the iPad, and they do an intake cardiorespiratory fitness test with an exercise therapist. And then for between 1 and 11 weeks prior to surgery, they exercise 3-4 times a week by Zoom with an exercise therapist. Every week, we send a mobile phlebotomist to their home to collect stool and blood. We have the tumor before and after exercise, and then we can evaluate all of the wearables and apply a whole bunch of fun data science and AI to understand the profiles of that. Everybody thinks that exercise trials are hard, and indeed compliance on them is 25%. These trials have 99% compliance because you ship a treadmill to people's homes and they're interactive working with an exercise therapist, and they're only like 10 weeks during the critical window after diagnosis when somebody is really excited to be doing something that might actually improve their outcomes. What does it do? It does all sorts of things. It obviously helps physiologically. We got patients, again, with 99% compliance to do treadmill running up to 450 minutes a week. You can see that improvements in blood pressure, improvements in body weight, improvements in body fat, trend improvements in muscle mass and percentage lean muscle mass, and small improvements in glucose tolerance. But you can also get really great data about what's actually happening. This is the resolution where we've monitored glucose over a 24-hour period for an individual. We can see what exactly happened during the window that they exercised. You can see the increase in heart rate, the increase in steps, but we now can map this picture to the 44 days that this individual was on trial and see the changes in different aspects of their behavior over that, and link it to outcome. And outcome, yeah, the tumors regress and we see improvements in molecular and other features. And I'll just give one sneak peek of what's going on. Here are a couple of patients as exemplars where we see striking reductions in immune infiltration and other inflammatory features as a consequence of exercise. Okay, so let me summarize everything I said. This is the patient journey that I told you, but I'm going to give you numbers. PSA test, $10 to $40. Digital rectal exam, $100. MRI, $2,000 to $2,500. Biopsy, $1,000 plus. If they do a deciphered DNA test, $4,000. And then treatment, easily $10,000. Real numbers. If we are able to incorporate genetics, we can reduce false positives and eliminate unnecessary tests all along this diagnostic journey. And by doing it early for a couple of $100s, you reduce the number of individuals tested at every step. So this is kind of what we just did. We talked, and all of these things happened for 630 men. To give you a sense, that was almost $2 million of healthcare costs that were incurred while I was talking to you, all before a single bit of treatment. That's why this is such a big opportunity for patients and obviously for commercialization. Thank you. I'll be happy to take questions in chat.
Sorry, nice job, Paul. And amen to the need for better, cheap information for patients and doctors related to prostate cancer. Your PSA numbers are spot on, and it's a huge problem that I saw at United Healthcare. Any questions from the audience? We have 10 minutes or so. Someone's on mute.
Yeah. Thank you for the presentation, Sarat from Hogue. It's interesting approach to this because it is everyone now for early detection cancer intercept programs, and your approach and exercise is really interesting. Everyone thinking about GLP-1 antagonists, all the other stuff that they can use in these setting. Any plan on doing similar, because ovarian cancer is next? And interestingly, in the guideline for ACMG guideline, they are recommending for all ovarian and pancreatic cancers to get germline genetic testing, but not for prostate. So we always struggle with approving these tests, but I think this will really help. I'm looking forward to see that publication come out to review the details of what somatic, I saw a little bit, it was a small print, TMPRSS ERG fusions you had on the top and so forth. So this is actually really going to help. So what any plan on doing on ovarian cancer?
Yeah, great question. I didn't talk at all about other cancer types to just give a clean, simple story, but we've done all of this in cancers of the cervix, cancers of the lung, and we are starting a significant colorectal study. Ovarian will be next after that. We just need the financial resources. But I can say that the compliance in cervix and lung is exactly the same in the 90 to 99% range, which suggests that there's a real model here for a way to automate and optimize this link of genetics, exercise, and the window between diagnosis and surgery. And it's literally, for most cancer types, the only thing that can be done because neoadjuvant is obviously not standard of care. Ovarian is a difference there, where neoadjuvant is actually fairly common. So it actually is a little bit more tricky than some of the other cancer types, and you can see why we started with the ones that we did.
No, great work.
Oh, and then you were asking, are we going to do GLP-1 and some of the chemical things? Not at first. I think that there is a sufficiently large appeal and market of you don't want to take a poorly tested compound, you've got 10 weeks, you are hyper motivated because you just got a cancer diagnosis. Therefore, here is something that on a per patient cost is $1,000 or less. It is very affordable, and yet it's something that people will be motivated to do and may have long-term health effects in all sorts of other ways. So yeah, we're thinking about keeping it a pure behavioral play rather than a mixed pharmaceutical or nutraceutical behavioral play.
No, that's great. It's the combination, especially on a population with BMI of 30 and over, to include that either gastric bypass surgery or some kind of interventions like that. It was a good data on endometrial cancer with that setting. So I was just thinking that it could be something with prostate cancer as well.
Yeah. I'll quickly note the cohort here had a median BMI of 29 and was sedentary. We've done other studies of non-sedentary populations, but all of these individuals reported survey data, less than 2,000 metabolic equivalents. So call it less than 2,000 steps prior to being enrolled in the study. So they are sedentary men between 50 and 65, basically. And it still had these benefits, which gives me a lot of hope for other populations. As you say, endometrial is a big target opportunity there.
Any other questions? All right, Steve, can I hand it back to you?
Sure can. Yeah, no, great. Thank you very much, everyone, for participating and your questions. And I will send a follow-up email with the emails of all the researchers, and even Dr. Rothman, who couldn't be here today, but he worked on some very interesting things. If you want to contact him, we'll have a description of what he's working on. But for now, thank you. Enjoy the rest of your days, and we'll be in touch. And thank you. Thank you, everybody. Thank you, researchers.
Thanks, everyone.
Thank you. Thanks, everybody.
Thanks.
Thank you, everyone. Bye-bye.
Bye-bye.